This is a quick demonstration on how to calculate the degeneracy of a DNA sequence for a given protein sequence. In other words, how many different DNA molecules could encode for that protein sequence. So let's say we're given this following sequence here, YPCQTRF, and we want to work out its degeneracy. In other words, how many different DNA sequences could encode for this protein sequence. Now to work this out we need a codon table. I just happen to have one here, and then all we do is we look up each amino acid and work out how many codons could be used to encode that one amino acid. So we start here with Y, we look up Y, which is tyrosine, and as you can see that's 2 codons, so we put down a 2. Next we look up the P, which is proline. And for proline, as you can see, that is 4 different codons. From there we go on to cysteine. We look at cysteine, and as we can see that is 2 codons. We then move on to glutamine, Q, which again is 2 codons, so we put in 2. We then move on to threonine, which is T, and as we can see here in the table it's 4, so we write down the number 4. We then move on to arginine, which is R, and as we can see here, this one has 6 potential codons that could encode for that arginine. So in this case we've got to write down 6. And finally in this sequence we've got phenylalanine, which when we look at the table is another 2, so we put down 2. Now from here, to work out the degeneracy, all we do is we multiply these numbers together. And when we do that, we find that we get an answer of 1,536. So that means there are 1,536 different DNA sequences that could encode for this short peptide sequence. And that's it. That's degeneracy. That's how we calculate it, starting with our amino acid sequence, working back through the number of codons, multiplying up the number of codons to give us the final answer.